gonna do some abdominal exercises now. Ah! What is that? Oh, 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 what the heck? That's disgusting. Oh my god, I got pooped on by a parrot and I turned into one.
That's not right.
Oh, I feel so sick. Oh, I'm so cold. I have the chills. But wait, I'm so hot. Oh, I'm sweating. I think I have a fever. Oh, and my head hurts so bad. Someone grab me a trash can. I might puke. Oh, my chest hurts. I'm having chest, liver, and or spleen pains and inflammation. Oh, that light is so bright, and I have a sore throat, and I'm so tired. I think I'm exhibiting flu and or non-bacterial pneumonia symptoms. Oh.
Okay, but anyways, I heard you have a lot of symptoms that sound like psittacosis.
Huh?
Yeah, you know what causes it is the bacteria Chlamydophila psittaci. It's an infection you get from birds. Have you been around birds?
One pooped on me on Monday. It didn't smell very good.
You inhaled it?
Yeah, I sniffed it.
Well, all bird types act as a reservoir. It's actually a good thing that I'm a biology major. I can go over the methods of introduction with you.
Yeah.
It looks like infection usually occurs when a human inhales the organism, so it had to have been aerolized from bird feces or from respiratory secretions.
Oh.
I mean, you could have gotten it from mouth-to-beak contact. Had that lately?
Not since a couple months ago.
I mean, you could have also had it from bird bites or handling the plumage of infected birds.
Could I have gotten it from, human to human?
No, there's no human-to-human infection. Since I am a biology major, I can go into the biology of it with you.
Okay.
Here's the pathology of Chlamydophila psittaci. Once inhaled, the Chlamydophila psittaci attaches to a susceptible epithelial cell in the lower respiratory tract. Some Chlamydophila psittaci may also spread via the circulatory system to infect the liver and spleen. However, we will focus on the lungs. Chlamydophila psittaci is an obligate intracellular pathogen, which means that it will avoid host defenses by entering a host cell. It enters this host cell by a mechanism not yet understood that could make use of the OMP2 protein. Also by a mechanism that is not yet understood, vacuoles containing the elementary bodies do not fuse with lysosomes. Still contained in a phagosome, the elementary body will enlarge and divide. When ready, it will lyse the cell, releasing the new elementary bodies. The cell being lysed causes damage to the epithelial tissue. This will cause host defenses to rush to the scene, and in turn cause inflammation. This inflammation will fill the lungs with fluid, the main characteristic of pneumonia.
Oh, that makes so much sense. How long do you think I'll be sick for?
I can't tell you. I think you should go to a doctor.
Oh, dang it. Do you have any recommendations of really good doctors?
I do. I know this fantastic one. Her name is Dr. Pepper.
Hi, I'm Dr. Pepper.
Hi, Dr. Pepper.
It seems you may have psittacosis. Let me check this book by my favorite author. It doesn't seem to be in it. Gee, how convenient. Thanks. Let me show you this video that explains how a medical microbiologist may go about diagnosing the psittacosis antigen.
Alright, so the main two microbiological methods for the diagnosis of Chlamydophila psittaci are the complement fixation test and the microimmunofluorescence test. The microimmunofluorescence test is preferred over the complement fixation test, so that's the one we'll be talking about today. Let's take a look at some symbols I'll be using in my explanation. Symbol number 1 is the antigen. Symbol number 2 is the antibody. Symbol number 3 is the immunofluorescent tag, also called the fluorophore. And symbol number 4 and 5 are just a positive result or a negative result. So how this diagnostic procedure works is a sample, in this case a sputum sample, will be collected. Then the sample will be prepared for the test by removing some proteins and then inserting control proteins so that the tester knows exactly what they're dealing with in the sample. Next, a sample of the antibody labeled with the fluorophore would be inserted into the sample. Now we can go over what a positive and a negative result would be. If the patient was truly afflicted with Chlamydophila psittaci, then the sputum sample would contain the respective antigen. So, when the tagged antibody is inserted, because of the immunofluorescent tag, when the antibody, antigen, and the surrounding proteins form a complex, that would then fluoresce or emit light, indicating a positive result. In a negative result, there would be no antigen to bind with the tagged antibody, so, well, nothing would happen.
It seems you do have psittacosis. This disease has caused inflammation in your lungs and inflammation, congestion, and other major organs such as the spleen and liver. It doesn't seem like it's not a contagious disease, but we'll put you through a 10 to 14 day treatment regimen to get you feeling better.
What are you prescribing me?
I'll be giving you a 100 milligram tablet of doxycycline, and you'll take that twice a day to stop the bacteria's protein synthesis by binding to the 30S chromosomal subunit. Or, for those who are pregnant or young children, I'll give them a 500 milligram tablet of erythromycin that you'll take every 6 hours to stop the bacteria's RNA-dependent protein synthesis. You're not pregnant, are you?
Do I look pregnant?
We'll go with doxycycline.
How long will I be sick?
You can be sick for the next 10 days.
Oh my god, I need to study for my med micro exam.
The symptoms wear off in a couple of days. You'll be fine.
Probably not.
So,
Is there
anything
anything
else?
I can do to prevent the spread of it?
oh, you could, if you have a pet bird, you can clean its cage a lot, make sure if you get a new bird, it's isolated from other birds, stuff like that, and don't go around sniffing poop anymore.
Alright. Thank you.
Nice to meet you.